Muy buenas tardes, estimados docentes, alumnos, personal administrativo, invitados. Sean bienvenidos a la presentación del Seminario Departamental, que en esta ocasión estará a cargo del doctor Eliezer Louzada, invitado del Laboratorio de Biotecnología Experimental. Nos comparte el tema What do I do with the tools that I have? Entonces, me permito ceder la palabra al doctor José Luis Hernández Mendoza, jefe del Laboratorio de Biotecnología Experimental, para compartir una breve reseña académica del doctor Louzada. Adelante, doctor, por favor.
I follow in Spanish. One moment. Bueno, el doctor Louzada es un profesor que trabajó para el Citrus Center de la Universidad de Texas A&M, Kingsville. Él estuvo ahí desde 1997 hasta el 2022, trabajando sobre el mejoramiento genético convencional y no convencional en cítricos. Y en los últimos años trabajó lo de transgénesis en cítricos. El doctor acaba de hacer junto con otros profesores de la universidad, acaba de hacer una publicación que es la última que ha salido en estos días, que es el primer reporte del virus de Citrus A en Texas. El doctor Louzada es de origen brasileño. Allá estudió su licenciatura en agronomía y luego después una maestría en ciencias. Posteriormente se fue a la Universidad de Florida, donde obtuvo el doctorado y donde trabajó algún tiempo ahí. Y en estos últimos años fue el profesor responsable del Laboratorio de Horticultura y Biotecnología del Citrus Center de la Universidad de Texas A&M. Hace un mes y medio, algo así, me tocó la suerte de ir a visitar el Citrus Center justamente el día que iba a tomar su retiro y me tocó platicar con él y por ello justamente me permití invitarlo el día de hoy, porque después de todos estos años creo que tiene muchas cosas que puede compartir con nosotros de una gran experiencia. Aún y que se ha retirado digamos, de la parte académica, sigue trabajando en una empresa privada sobre el mejoramiento genético de cítricos. Así es de que, a continuación el doctor Louzada dará su conferencia. Dr. Louzada,
yes, sir.
now the microphone is yours.
Okay, thank you so much. Do you want to give me time for a translation? Do you want me to go ahead and try to speak in Spanish? My Spanish is very poor, but I can try. Whatever you folks decide. Well, everybody speak okay with English.
Speak in English, don't worry.
Okay, thank you so much. Thank you so much. Yeah, I decided to talk about these tools that we have because we accumulate as we go through our master degree. And like for me, I started basically in science my first year in college. So they called me lab rat because I was in the lab all the time. So I started working with soil science. I was in the lab of soil science, physical chemistry and soils. And I really enjoy investigations. So I did my master degree now before the reason why I decided to talk about this because we get all these tools as we go studying and participating and work in the lab. And suddenly we land a job that now we start our own lab and we have all those basically training, all those technical expertise in a toolbox, but now we start our own lab. So how will we go from there? And so I'm gonna give my experience because when I started the Citrus Center, I came from the University of Florida. I did my PhD over there. I started in Brazil and I finished at the University of Florida. I started in 1990. And so I had the opportunity to working a lot of different techniques. So sadly, 7.5 years later, because I worked over there also in the private industry. So, but now I was in a place, my own job, my first job. So I'm going to give an idea about, how I start, and how I could be successful in developing my program. Okay, I had my bachelor's in agronomy. This was 1975, a long time ago. And I started undergraduate research very early in college. And I did my master degree, and I finished in 1978. I started in 1975, so it was 3 years. It was a very, very hard work there, because was harder than my PhD, I would say, because I was developing something that was not there yet. So I have to basically come with a procedures and develop my own methodology to study the residue of herbicide picoran in 2 soils over there in Brazil. It's like, if you know the United States, you have a California, you have a Florida. So, and I was basically comparing, I was in Texas, so I had to fly to one a field trial to another field trial, collect sample, and harvest soil up to 120 centimeter deep. So it was really, really very hard. So, and then bring this to my city and work in those soils, and I use a bioassay. So it was extremely, extremely hard. And after that, I had to, when I finished basically the research, I started writing. We didn't have computers, so had to pay somebody. Basically, I had to write by hand and pay somebody to basically type for me. And then when I had a very heavy statistical analysis, was a split plot, my experiment, so it was a factorial. And I could not do by hand, so I had to use one gigantic computer over there, punch card, hours and hours at night, punch card, to be able to do my statistical analysis. So it was really, really, really hard. But it pays off, because I learned all of that. The most important, to have persistence. It basically, it prepared me for everything that I had to go after that. So after that, I work in private company, most in agronomy. I work with soybean, I work with wheat and a few other crops. And 15 years later, I decided to go for my PhD. And I decided to do genetic and cell biology. So I start in 1990. I got in Florida in 1990, and I work with somatic hybridization. I do also genetic transformation. So I was 7.5 years working in Florida. I was, as soon as I finished, well, before I really finished my PhD, a company, private company, citrus company in Florida hired me to develop a mandarin conventional breeding program for seedlessness. So I had basically, I was responsible for the whole breeding program there. And I was able to make almost 3,000 triploids, develop almost 3,000 triploids using conventional breeding, just exploring something that I found that was really, really a blessing to my program there at this private company, because I found that one of the mandarin variety produce two-N gametes. So it was very interesting, because I could use this mandarin that produce diploid gamete and breed with another one that had conventional haploid gamete and produce many, many triploids. Some of those, about 20 of those 3,000, they were already patent. So from there, I came back for the University of Florida, because I want to continue my career in science. And so I was constructing a genomic library of citrus for mainly physical mapping. So And 7.5 years later, I was hired by Texas A&M University at the Citrus Center, Texas A&M University, Kingsville, the Citrus Center. And that was the real beginning. So now I get in there is a commodity-based center, a research center. So that works specifically with citrus and with the citrus industry. So I have a mandate that was to use whatever tool I had to solve the problem of the industry. They want a new variety. They want a variety that's resistant to a set of diseases. They want some that can reach the market easier than the consumer like it. So, and they are very interested in rarer grapefruit, some that was much rarer than the one that they had, and they had better characteristics for the industry and then for the consumers mainly. So this was first mandate. As soon as I was hired, they told me, well, we need new varieties, and this is what we want you to do. So you need to use our tools for you to develop new varieties. So I had all that toolbox full of techniques that I learned before, but now I had to develop first a vision. I had to develop a vision where I want to go for me to be able to go. So all those techniques was very important, but without a vision, they would not work for me. So, but when you work in the university, which is different from private industry, I had the opportunity to work in private industry, and now I was in a university. Is very different. When you are working in the industry, they have already established goal that the industry want to reach. And that they have some tools and they want for you to apply those tools, and they provide a lot of the program. They have already the program. When I get to the university now, I am the boss, basically. Yes, I have a director, but really, I develop my own program. The director has no influence in my program. So now I was 75% research and 25% teaching. This theoretically, because I had much more than this. I had to teach one class per semester. I had to publish in peer-reviewed journals so that I could be promoted. I have to serve a committee. I have to write grants. I have to get money to be able to start my program. And if I was successful 5 years later, 5 years later, I would be promoted and receive a tenure. So this was the first thing that we are interested in, get our research done so that we can get promoted and get tenure to associate professor. So basically, when I started, this was the state of the science in the United States and around the world. First, to purify a plasmid, we didn't have this wonderful kits that in 15 minutes we can purify a DNA extremely easy. So we need to use a cesium chloride, a bromide gradient that take almost 1 week. For you to have an idea, the human genome in that time, they were just in the first draft, and they had a $300 million already spent in sequencing. We tried to, I'm going to say we, because I was part of the steering committee for the citrus genome. And we were trying to put a white paper together so they could get funding to have the citrus genome sequence, the whole genome. But it was so expensive that we had to have a consortium made of 3 countries to be able to have. Today, I want to show you something that I'm going to put this down a little bit. Hold on just a second. To show you basically the situation today, just for comparison. Okay. I don't know if you have heard about Minion. Okay. This is an amazing really tool today. I want for you to look.
Dr. Louzada.
No. We can hear you, doctor.
We have a trouble.
You cannot hear?
Okay.
Yeah.
You cannot hear?
Yeah, it's okay, doctor.
Now it's good.
Could you see the video and hear?
No, the next slide.
Can you? I can put in here. I don't know if you can see it. Can you see better this way? Can you see this way? What's the best way? Full screen or?
No, it's okay.
It's okay?
Yeah.
Oh, I'm sorry. So let come back in there. Sorry.
If you go to next slide.
We can hear you, doctor.
Okay. I don't know if you could see. I want to show you that what we did, before for more than $25 million. Now you can buy this equipment. Can you still see my screen?
Yes, sir.
Okay. So what we did before was so expensive to do. Now you can do this equipment is a palm size and it costs $1,000. So it's amazing what you can do today. So if you start today with the tools that you would have, it would be amazing. Now, when I started, I didn't have all of this. So basically, this was my lab when I started. Was basically not a single piece of equipment. Was just a classroom that they told me, well, this is where you're going to build your lab. So I didn't have a single equipment. The City Center at that time, they would do more field research. There was not any kind of labs really ready. So there was one in plant pathology with a few equipment, but nothing more. So this was my lab. And then from there, I would have to, in 5 years, have enough results for me to be promoted. So this is almost insane to think that I had to build everything in 5 years, have enough publication out of my lab for me to be promoted. So, and this is how I started. So about 3.5 years, I had basically my first lab in that place over there, that empty room. I had my first lab. I didn't have a big startup fund. I got about $25,000 to start my lab, but we had to build everything. When I hired my technician, I just had a drawing in the bottom in the floor of how I would build my lab, what would be there, and I was just a planning. So I had to have the vision what I was going to make. So, and then this is the other part of the lab. So we had now a very well set up lab, and this was about 3.5 years later. And then I started involving students in my research, lots and lots of students. I started a program in the year 2000 to bring students, undergraduate students, to provide undergraduate student research. And basically is how I start my lab, by getting grants so that we could now set up the lab and be able to get now research done. And then later, about 2.5 years later, we start another lab, the one that you see here, and then basically now had a lab complete. It took about 5 years to have a really, really complete lab, but we could generate enough research from there. From that empty room, we could start our research, and then so 5 years later, could get promoted to associate professor. Okay, these are the mandate based on the industry. They want new varieties, and they want more options. So we start looking for methodologies for us to work. So we decided to use mutation, because citrus, a lot of mutation happens in citrus. So we decided to induce mutation. So we start working with induced mutation, also looking for natural mutation. So our first variety was the number 2. Okay, this we call Texas Red. This Texas Red, we basically was a natural mutation that we end up bringing from the field to the lab through tissue culture, and from there created this variety. So this Texas Red is patented now, is available to the growers, is also available in South Africa. They want to license, so they already have a commitment to with the university to be able to plant this variety. Also Spain already have this variety over there. The number 1 is also a grapefruit, but it's a grapefruit with a little bit of flavor of mandarin, very different flavor. So this was, we just call golden grapefruit because of color. There's not any baptized name yet. It's not patented yet. The number 2 here, the Texas Red, you see in the side, the yellow one is the conventional grapefruit that we have today. The side, Texas Red. The number 3 is a Maya lemon that we call here Valley lemon. The difference is that the normal lemon has a lot of seeds, so we made one that's completely seedless. But you see the number 4 is a chimera. What we did with this chimera, you see one side very yellow, the other side very red. So we went inside, got some tissue from this part, and the tissue culture could recover a plant that produced this red color, which is different than the number 2, okay? So the number 4 is different than number 2. So, and it's redder inside than the number 2. So this is basically what we put together to bring, because the growers, they want to see things, they want to see results. If you don't have the results, they don't give you the support. So we had to start using simple techniques to be able to show the growers, yes, we can do, and we are going to do. So we start with a simple technique, not using our toolbox, but basically using simple techniques to be able to do this. We also use a lot of conventional breeding to produce some mandarin hybrids, seedless mandarin hybrids. They are still, they didn't fruit yet because they take a long time, up to 15 years, to be able to produce fruits because they are very juvenile. Very juvenile. Citrus has a very long juvenile period, which is very complicated for breeding. So, but we have several plants, potential hybrids, potential triploids that is going to be evaluated by the person who take my place in there. This is the Texas Red that I showed that is already patent. Okay, this is already available to the growers. We also, another thing that the growers was asking, they need phytophthora resistant sour orange. And is a serious problem here in Texas because we use our irrigation system is a very old system. It's just that we flood everything. So with all this flood, we have a lot of Phytophthora, and we have a lot of dead tree because of Phytophthora. So we decide to using a very simple technique to be able to develop a Phytophthora-resistant sour orange. What we did is use selection pressure. We got about 5,000 seeds of sour orange, and we germinated this in a soup of Phytophthora. We produce Phytophthora colonies. It's basically pure culture Phytophthora, and we prepare a very, very heavy soup of Phytophthora, and we put the seedlings to grow. We cut the roots and put to grow in Phytophthora. And what happened is that only one survived. After this, we select this as a Phytophthora resistant. We test several times. What you see here in this picture is exactly here is exactly the Phytophthora resistant. You see that under very high pressure Phytophthora, you see the root system is perfect. So this we are starting now a field test to make sure that they can really be in the field as resistant and as they were in a greenhouse. So we are now doing this, even though I'm not working there anymore, but I'm still interact with not the person that is hired yet, because it's still going to be hired, but I'm going to be interacting, guiding them in doing all these field trials. One other thing that we did, we decided to use a technique that in that time was very, very common, differential display. And using differential displays, we isolate a few genes. One of them that we call CSM1 for calcium signal modifier. This is a cyclic nucleotide gated IO channel that transports calcium inside from the outside to inside of cytoplasm. And we found that this gene to be very interesting because it modulates the calcium signal. And this causal signal, they vary from one to the other in amplitude, in frequency, duration, so that these signals are sensed and detect the protein, decodify based on this decodification system, sense and detect the protein, make a kind of shotgun approach that triggers several genes, including a lot of disease resistance. So we decided to use these genes in genetic transformation. So we use agrobacterial transformation. We develop, we basically set up our system for agrobacterial transformation, and was very efficient. We got very high efficiency with the strain that we have. And so you see here, I'm sorry. The pointer. So you see here, the cambium cells, you see the transformation here, the cambium cell. Exactly these cambium cells are the one that are more totipotent. They really very quickly regenerate into plant. You see here, one shoot coming. Okay, so it's very efficient, and we got very high frequency of transformation. So we regenerate several plants, and we test several of these plants. We got orange, grapefruit. Like these are grapefruit, ruby red. This is non-transgenic in here. This is transgenic in here. What we did, we inoculate the Phytophthora, was Phytophthora inoculation. And this is what happened to control. This is what happened to transgenic. So the Phytophthora could not infect it. So we make another test. We got the Phytophthora soup, and we put some leaf discs. You see what happened here with the control. You see what happened here with the transgenic. Nothing. So it gave basic immunity using this gene. Okay. Another project, this was basically the first project that we started at the Citrus Center. Our first basic purchase was an ultracentrifuge, and we had our system to work in tissue culture, and we started using this methodology, that's microprotoplasm-mediated chromosomal transfer. So what we want to do, we want to produce some citrus plants that have one or two chromosomes from other species. So we're trying to transfer like cells, chromosome from orange to grapefruit and vice versa, just to add a few characteristics. So what we did is we produced embryogenic suspension cells of several species, and after we synchronize the whole cell cycle, because we need to have this embryogenic suspension cells in very high mitotic activity. So we produce these embryogenic cells, and we synchronize the cell cycle, use hydroxyurea. And after that, you depolymerize the spindles using amyprophos methyl. And then the chromosome was scattered around the cell, completely scattered, because they didn't have the spindle anymore. So you see here, this one that had the chromosome scattered. And then after the chromosome is scattered, they start decondensate and form a membrane around themselves. So now you have a nuclear membrane around each chromosome. So they have, these are a multinucleated cell, each one of these one nuclei. Some of them with one chromosome, some with two chromosomes. So it's basically you have the whole chromosome surrounded by nuclei. After this point, okay, now we use a very high speed centrifugation. Is the range of 120,000 G, and this is per call gradient. We now using also one specific product called cytosalasin B that destroy all the cytoskeleton. Now, each one of these nuclei, when you go through the high-speed centrifugation, they come out of the cell carrying a little bit of the membrane. So they circularize around each of the nuclei. And so they make microprotoplasts. It is about the same system that they use in mammalian cells for microcells, okay? So now having here in these slides, this picture, you have here the one protoplast, and here different kinds, different sizes of microprotoplasts. This after the centrifugation, we collect this band. You have another band in here, another band in here, and we pass them through a series of sieves. The lowest sieves is 5 micrometer. So that purify out of the debris. So now we have some very small microprotoplasts. You see here, this is a very high magnification. You see here, only one. There's only one basic chromosome. We have here, you have 3. So now we make a fusion of the protoplast with the microprotoplast. So you see here in the bottom, in this part here in the black white, you see a protoplast in here. You see a microprotoplast in here. And then after polyethylene glycol, they start getting close, and you see now that the protoplast basically swallow the microprotoplast, and now you have one single protoplast in here with this part in here is the microprotoplast, and the remaining are the protoplasts. So This way, we were able to regenerate not too many plants, but a few of them. And like here you have, if you count the chromosome here, you have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21, 22, 23, 24, 25 chromosomes in here. So it's basically is an aneuploid. We got also some with 1 and 2 chromosomes. And this plant, something very interesting that happened is that this plant, it would grow to about 3 feet and would die. So we maintain for the longest time, and that all of them, they would grow but die out, grow and die out. We would take some buds and graft again. They would grow well, and they would die. We couldn't figure out why, but the methodology are very interesting. But I think because the cytoskeleton, when we prepare the microprotoplast, the cytoskeleton destroy in some way affect the final product. So, but we were able to produce a few plants out of this, but not something that we could say that is going to be something that's going to come ever to the market, or that is going to be anything good come out of this. But was an attempt that we had to be able to transfer chromosomes. This could be interesting, even if we want to use some of this microprotoplasm for sequencing. Probably would be interesting to use this as a tool for another technique. We decided to quit this procedure because it takes about 1 week for the whole procedure. From the point of preparing the cells and prepare high mitotic activity cells, all the way for you to be able to fuse, it takes about 1 week and several manipulation. So it's very easy to get contamination. So we saw that was not interesting in terms of what to expand to have the technique and the final results. Because of this, we decided to stop. This is just a view of the microprotoplast solution. We also had a lot of transformations, genetic transformation done, and we decided to make market-free transgenic plants. I know that the market, the consumer, they don't like to have a marker in their plants. You know, the problem with genetically modified plants, if they have genes over there that are genes from virus or from bacteria, this is a big problem for them. So we decided to make a genetic transformation, but eliminating the markers. So we decided to use one technique where we use a dexamethasone-induced recombinase, so that we have, this is just our first test that we did it. We had one cassette here that have half of the promoter in the side. Let me see if I change this pointer in here. So we have half of this promoter on side, the other half in the other side. Okay. So, and I had here a recombinase site in here, another recombinase site in here. So one here, one here. This is a recombination site. And I have my recombinase gene in here. So, and this is induced by dexamethasone. So when I introduce the plant by agrobacterium, I had in here my 2 markers. This is a negative selection, this is a positive selection, and I have here the recombinase gene with the inducer. When I introduce the plant by agrobacterium, I treat with recombinase, and then the recombinase eliminates all of this from one side to the other, and then you have now the promoter. They have the half promoter, the other half promoter with a cherry, and now we have the complete reconstitution of the Mario cassette. So what happened that we have the M-cherry gene inside. So now I can have my plant expressing M-cherry. So I can look in the microscope with a fluorescent so I can see how many plants I obtain that express M-cherry. Okay. So this negative selection, I'm not going to go in detail now, but basically plants that still have the marker will die because of this code A gene. It produces a toxic compound in all plants that still have the markers, because we treat them with 5-fluorocytosine, and this code A, it transforms 5-fluorocytosine to uracil and kill all the plants that still have the promoter. That still have the markers, and this way produce marker-free. We, for our experiments, use another cassette that is the one with the number 2, and here you have the excision sites with the enzyme. We have here here is the recombinase. We have code A, the negative selection. We have the NPD2 positive selection. You have isopentenyl transferase in here that produce a cytokinin. This will improve regeneration, and was fantastic because I could get plants like I could never obtain before because cytokinin produced by isopentenyl transferase. And then M-cherry also here for us to make the selection, visual selection. And then we have in here now our gene of interest. Okay. Here our gene of interest, CSM1, that is the second nucleotide gate ion channel. All these genes, they are with plant promoters, okay? We test about 16 different plant promoters for us to be able to use with all these genes. All the genes are driven by plant promoter. So after this is just a view of the full plasmid, okay? Now, what happened, you have here in the top, okay? Here you have here expression of M-cherry. Here you have expression of M-cherry, and see, you don't see any plant over there because they are not expressing cherry. So I just put a lot of light in the bottom one so they could see what cannot see in the top, okay? So you see now that even though you have the bottom one, the bottom part, will have, these are embryos, okay? The embryos, they express M-cherry, but the plantlets are not expressing M-cherry. So the shoots, they don't express M-cherry. So this way you could select a large amount of plants that does not express M-cherry out of embryos that express M-cherry. So we could produce several plants not express M-cherry. We check for the presence of the genes. The top one here, this one is for the A is M-cherry, okay? You see this, the N19 is expressed M-cherry, P16 is expressed M-cherry. You have here in the B, this is a code A, that code in the amylase. You have this that is expressing, okay? You have this that is expressing. And now you see the ones that does not have, you see this, N18 does not express M-cherry, does not express code A, but express CSM1. You have here all of these guys in here. They don't express M-cherry, they don't express code A, but all of them express CSM1. Same for P1 and P2. And then these are the negative control here, and this the positive control that is the plasmid. Okay, So this way you could obtain several plants expressing that does not express cherry or code A, so they don't have any promoter, any markers, okay? All the markers are eliminated. We check also for NPD2, they're not there. We test for isopenyl transferase, they're not there, but they have the CSM1 gene. So we have these plants now in the greenhouse, and we are going to start testing them for Phytophthora and for HLB. We have a few plants in the field, probably about, I would say, maybe a quarter of an acre in the field that you are testing for HLB. Now, the most exciting part of my career was to work with the students. This was the most fantastic. I started the program in the year 2000, and I was funded from the 2000 all the way to my retirement, basically, to work with students. So we had a high school student, undergraduate student, you had a student from 2 years college. So we established a very, very good undergraduate research and work with high school students, so that we had the summer camp high school students, science teachers. We had undergraduate student coming, and they were recruited to graduate studies. We had a student from 2-year college that ended up being a recruitment tool for our university. So this program was the closest to my heart. I would say that from everything that I did since I was hired, this is really the main thing of my career. Why? Yeah, because of the technology that we use, all what we do in terms of publication and everything else, it's going to be forgotten, no doubt about it. But what happened with these students is fantastic. We had, out of the undergraduate program, 15 students that went all the way to PhD. So I have today several professionals in biotechnology companies, in the universities. At the Citrus Center, I have 2 of my students at their faculty today. You have a student in Ivy Leagues, universities, and all of this because of this undergraduate research program. So when this is a hands-on research. That's the program that I established. Is a very, very beautiful program. This all undergraduate students have my technician there. All the others are students, part of my lab. So what I saw is that they brought so much energy to the lab. They basically changed my whole perspective as a faculty. They were basically the cheerleader of my program, because they were the ones that were responsible for my success in research. Because they will do the job, they will do well, they were well trained, and I could see them going all the way to PhD and being professionals. And the most gratifying thing that happened to me, see, this is my lab in a program that we had at the Citrus Center. All of these in this table were, in that time, students in my lab, okay? And then when I retired, it was fantastic to see all these students there. They are professionals today. We have here is a faculty at Citrus Center. There's a faculty at the Citrus Center. We have all of them are professionals today, work in different areas. And I didn't know that. They made a surprise for me, and I made this dinner there. And they invite all the people, all the students that were far away in other universities. So by Zoom, I could see like this one that today a very high position in Washington, D.C. at USDA. This one is a professor in Ivy League school. This one is a very high position biotech company. And all the others, this one is a Bayer company. So this one got his PhD also, and worked in San Antonio, Texas, at the university over there. So for me, this was the most gratifying thing. Yes, I got a lot of results, I got a lot of publication, I was promoted to full professor, and I did a lot, but nothing compared to the beauty of a these students going their career, and these students would never have opportunity. I always talk about this student here. This student here, he came into my lab as an undergraduate student, extremely shy. He couldn't even speak, basically. His father came with him to ask me to allow him to be in my lab. He was an undergraduate student at that time. And he was going to college at that time. And this young man became a fantastic scientist. Today, he has a very good program in Ivy League University. And it's amazing because all of them had no idea about science, would never have this opportunity if it was not coming to the lab and be trained in the lab. So this is why I say that this was the most gratifying part of my career, is to be able to help students reach their goals and be professionals in several areas. And all of this, they helped me to build my program. They helped me basically, their success is their success. So they gave me this, and I thank them a lot. So, and if you have any question, I'd appreciate to answer.
Very interesting. Your life.
Let me.
Jóvenes, ¿tienen alguna pregunta para el doctor?
Oh, I'm sorry. Let me just take it out of the. Any question?
In the chat.
Jóvenes, ¿tienen alguna pregunta? Any question?
Yes. Hold on.
Dr. Louzada, as you look the question, or you need a
Say again. I'm trying to remove my screen here. I don't know what's going on here. That it's not giving the full screen.
on right corner
Okay.
down?
Okay.
Have you worked on abiotic stress on plants?
Yes.
This is the question.
That's okay now. I think it's okay now. So yeah, if you have any question, let's see.
Have you worked on abiotic stress on plants?
Have you worked in what?
Abiotic stress. Abiotic stress on plants.
Okay. Okay. Have you worked on abiotic stress on plant? Okay, I had one gene for abiotic stress. It's very interesting gene, but I didn't have time really to work with that. It's a gene from rose. they call extreme drought tolerant gene. And these are from Rose. They use in Arabidopsis, and this gene really was more for drought. And this gene, basically, they had Arabidopsis thaliana with 21 days without water. And when they put water, that little guy came back to life, basically. So this is what they call extreme drought tolerance. I didn't have a chance to work on the gene. I had the full length of that gene, but I didn't have time to work with that because I was committed to some project, mainly with the California citrus industry. And because of that, I could not really work with that gene. So, but I hope that someone finds some time to work with that gene because the literature at least says that this is very, very interesting.
¿Tiene alguna otra pregunta? Any other question?
I don't know if the person understood, so if you want to translate to them.
No.
Tienes un gene de Arabidopsis que es de rosa, okay? Y que este gene es llamado extrema tolerancia a la seca. Es un gen que es ponerlo en Arabidopsis y esta Arabidopsis es ponerlo 20 días sin agua. Después de 20 días sin agua, eles puseram água e a planta sobreviveu. Entendeu? Então, é uma extrema tolerância à seca. É um exemplo de flor rosa.
Leonardo, sí, la otra cosa que dijo es que eso lo obtuvo ya muy avanzado su tiempo de trabajo y que ya no tuvo tiempo de probar eso y que lo va a ver con la industria de California. Well, I agree with you that working with the student is one of the best reward.
Yeah.
When we look the face, oh.
Yeah. So if you have any other question, if you want to communicate with me by any chance, Jose Luis, you have my email. You can give an email if anybody wants to ask any question, if you have any interest in know any other thing. I had several other projects that I was involved with. I was involved in project also with CRISPR, but just give an idea about some of the projects I was involved because of the time. So.
Yes, I have your email, and the next time I think we need to talk.
Sure. Yeah, no problem. And it's a pleasure for me to be here. I really, really enjoy and interact. And now that I'm retired, I'm going to be a little bit farther from, I'm going to try to maintain my contact, but it's not always possible. So I appreciate the fact that you invite me for this presentation. And I'm really excited about this. And so thank you so much for this opportunity.
We are excited too. Victor?
Well, yeah, we have in Spanish reconocimiento, doctor, for you.
Recognition, award.
Yeah.
¿Cómo?
We can have a award for you.
Well, yeah.
Okay.
Okay, let me do it. And it, oh, no, let me see. Está. Ok. You see? Do you see? Ok. I read. Dice, Instituto Politécnico Nacional, Centro de Tecnología
Okay. Yes, yes,
Económica.
yes.
Otorga el presente reconocimiento al doctor Eliezer Louzada por su participación como ponente en el Seminario Departamental Virtual del Laboratorio de Biotecnología Experimental. What do I do with the tools that I have? 28 de noviembre del 2022 a través de la plataforma web del Centro de Tecnología Genómica. Atentamente, la técnica al servicio de la patria. Doctora, lo firma la doctora Amanda Alejandra Oliva Hernández, la directora del Centro de Tecnología Genómica. We send to you,
Thank you so
doctor.
much. Thank you so much. Yes, thank
Okay.
you.
We send to you.
Well,
And if
we have
any
a
questions,
recognition.
Citrus also, I'm very involved now in basically in growing citrus. So I'm available anytime that all of you, any of you need some information on growing citrus. Okay. Thank you so much.
Thank you.
Thank you, Dr. Louzada. This recognition I will take later. The next week, I go to Citrus Center.
Okay. Yeah, I
Okay.
usually go there every 2 weeks, sometime. If you let me know, I may be there. I have grows in Waveco, so I go there frequently in Waveco. Okay.
Well, thank you very much, Dr. Louzada.
Okay. Thank you so much. Okay.
Happy
Bye-bye.
July.
Okay.
Bye, everyone. Okay, bye.